So without further ado, our next session that we're going to move into is going to be current and emerging clinical trials for CMT. For this, we will hear briefly from 4 speakers. Each will tell us where their company is with developing clinical trials. So we're going to do about 5 minutes. Again, this is just to give an overview and to start a little bit of a conversation. So our first speaker is going to be Vera Kiyasova, Director of Clinical Development at NMD Pharmaceuticals.
Hello, everyone. There must be some couple of slides that we've sent to Susan a week ago. There is a clicker. No, I think it goes to the next session from Janine. I can certainly improvise, and tell about our trial. It's not a problem at all. Okay.
Let's start there.
Let's start there. So, good afternoon again. I'm Vera Kiyasova. I'm a neurologist, neuroscientist, and currently medical director at NMD Pharma. NMD Pharma, a small Danish biotech, 50 people, with 3 ongoing phase 2 studies in 3 different neuromuscular disorders, and one of them is CMT. And the intention today was to have a slide that shows, a little bit of a team and also presents to you what is this trial about, because some of you have heard our name, but we're slightly different probably to all the other therapists. So what we are working on is the tablet and oral medication, which is targeting specifically the muscle. And that's what is important. So in the field where we always talk about neuropathies, the way we are trying to address is by acting on the muscle. And Mary Riley presented it nicely, in a couple of sessions before, that, when the signal from the nerve that comes to the muscle is weak, you somehow need probably to help the muscle to be strong and contract better. And so that's exactly where our molecule work, acting on the CLC1 channel. And why as you also hear, we study it in different neuromuscular disorders, because we truly believe that there are several conditions where muscle needs help. So, and that's also the reason why in our currently ongoing phase 2 clinical trial, we've been enrolling patients with CMT type 1 and CMT type 2, which would sometimes be criticized, in the previous year, saying these are very different clinical phenotypes. But what we do believe is that independently on the genetic origins and the clinical phenotype, thank you very much. Here we go. This oral medication might be beneficial for patients with different types of CMT. So this was quite a short study for adults aged 18 to 70 with CMT types 1, 2 for the 21 days of treatment. Here we go. 21 days of treatment for a lifelong disease might seem extremely short. And there's a certain reason for why the trial was designed so short, but it's also the reason that the mechanism of action is quite fast. So we've shown it in preclinical models and in phase 1 studies and in phase 2A studies in the different other disorders, that the drug acts fast. And in all the types of muscles. So we do believe that in 3 weeks, we can see the difference. The objectives were for this trial to see if our molecule may help improve strength, endurance, and balance. And of course, due to the early stage of development of the phase 2 study, we are specifically looking at safety, because that's what we as drug developers and that's what regulators are expecting from us. So safety first, looking at the hints of efficacy. The way we looked at it was to measure the 6-minute walk test that you know better than me because you've been tested for it regularly. We've been using also the composite score of the CMT FOM, which, I mean, if we basically talk to each other, it's how can you perform physically during the testing? And of course, the patient-reported outcome. And also, I truly believe that it's important to mention, and we had a very interesting conversation with one of our sites yesterday. We added to this trial the wearable device, because quite often we hear from the patient communities, and especially some of you who participated in Next trial, you know what? We come on site for this visit, and that's how we perform. But in between this visit and that visit, there's entire life. And it's very different from on everyday basis. So having a tool, which is still exploratory, we're still in an early stages of development, I'm not over-promising, but adding a tool that may help us researchers to look at different variables and what happens in everyday life. This was also an important point for us, even, for this probably a short trial. And because I sent the slides a week ago, I didn't add a pop-up message that was considered to say that enrollment is now finalized. So this week, we had our patient number 80 getting into this trial. So in a sad note, it's unfortunately too late for some of you whom we meet here and who would be eager to join. But on a positive note, this also means that from now on, we finalize this, analyze the data, and hopefully in the beginning of the next year, we'll be able to share with the community, and we all hope for positive results, and then start planning for the later stages. And as we just discussed with someone in the public, hopefully opening to all comers and all types of CMT. So that's something that worked more or less before the slides arrived. And that's the company, that's who we are. Okay. Thank you.
Thank you so much, Vera. Next, we will hear from Dr. Evan Bailey, Senior Vice President of Clinical Development and Head of Medical Affairs for Applied Therapeutics.
Good afternoon, everyone. I'm Evan Bailey. I'm actually the Chief Medical Officer now at Applied Therapeutics. That's fine. I still wear all of these hats as well, so it works. So I think some or most of you are familiar with Applied Therapeutics and the work we've been doing in CMT SORD, which is a specific subtype of Charcot-Marie-Tooth for sorbitol dehydrogenase deficiency. Now, one of the challenges in CMT and the multitude of subtypes is finding a signal or a biomarker that could be a sign or signal of disease. Fortunately for CMT SORD, there is that biomarker, and it is sorbitol, which is an end-stage sugar alcohol. This builds up in the nerves and causes the progressive neuropathy that patients develop with CMT SORD. They're especially affected in their lower extremities. However, their upper extremities are also affected. And they do have the day-to-day burden of fatigue, pain, and just challenges overall. So we have completed the randomized double-blind portion of our phase 3 study. We presented some of that data at the PNS conference a couple months back. And right now, those patients who elected to stay within the trial are in the open label extension. So everyone's receiving the drug right now. It's an aldose reductase inhibitor, which blocks aldose reductase, which is the enzyme that converts glucose into sorbitol. Now, we're able to reduce sorbitol by approximately 40%, which is statistically significant compared to placebo. And we had a significant improvement on the CMT high, which is the health index. And for me, that's a really key thing because that's a patient-reported outcome. And in CMT, it's actually a patient-reported functional outcome. The majority of the questions in the subdomains actually focus on function and not just how you're feeling in terms of an emotional component, which is critical. However, to have relationships on how a patient's able to report their own functioning, to me, is exceedingly important. And then we can look at how that ties into actual functional measures over time. We do know that CMT is unrelenting and progressive. We had a signal on some MRIs that patients who did make it to the 24-month mark did look like they were having a slowing of the increase in bilateral calf fat fraction. With this, I say that because for ongoing clinical trials, if you have an open label extension, there are opportunities to gather more data. So we're in the midst of gathering more MRIs for patients who've been on treatment. We'll be able to analyze those and look at overall exposure to see if what we're observing is true, where in that second 12 months of exposure, there may be some slowing on that. Now, that's key because we've been able to show with other studies that MRI findings correlate with functional endpoints, and they're actually predictive of functional endpoints several years out, which is so important for a slowly progressive disease. So that's where we are with the current ongoing open label extension of the study. We are in the midst of conversations with the FDA and next steps. One of the potential pathways forward is something called an accelerated approval, for which you have to do a confirmatory study. We've been working on designing that confirmatory study. We've been working with our key physician experts, working with the agency, going back to the literature, and we've designed that study and are trying to plan sites. We don't know exactly when it'll go live. It should be coming up in the near term, but that is the next study. I think one of the biggest challenges that all of us would speak to is aligning with the FDA on which endpoint is meaningful. I come out of several rare diseases in my prior life, one of which is cystic fibrosis, which actually for CMT SOR, there are about 3,300 patients. So CF in the US, it's over 1,000-fold more. And they have disease-specific tools that are recognized by the FDA. And here we are with CMT. We actually have the CMT high, the CMT FOM, disease-specific tools that are validated and studied. And it is surprising how challenging it is to get the agency to accept that. And so I think one of the things that the patient community and all of us who are clinicians and physicians and researchers can do is to advocate for these disease-specific tools and to also provide support that these tools are applicable to CMT subtypes across the board and not just specific CMT subtypes. Because sometimes that's the feedback. Well, how do you know it's going to be relating to this specific subtype? You can present them with all the same phenotype, disease manifestations, and rate of progression. Still cross your fingers. So those conversations are ongoing, and that's where we are right now. We're very proud to be the first company to have a phase 3 program and be this close to a potential therapy for a specific subtype of CMT. And we're hoping that if we're able to get this across the line, it'll provide an impetus for other companies or other research funding to get other CMT subtypes the research approach and treatments in the future that they deserve. Thank you.
Okay, next we're going to hear from David Goldstein. Dr. David Goldstein, co-founder and CEO of Actio Biosciences.
Well, thank you. It really is a pleasure to be here with you all. Let me start by just making some general comments about running trials in rare diseases. It's a very challenging thing to do. It's very challenging to get companies to focus development efforts on rare diseases, and running the trials themselves are challenging. There's often a temptation in the affected communities and even amongst researchers to try to get to something as quickly as possible. And unfortunately, that just really rarely works. What does work more reliably is to really do all of the difficult background work necessary to have proper drug development efforts and to run appropriate trials. This takes a very, very patient approach to getting ready to run trials. So I would want to start by just acknowledging the approach that the CMT-RF and the broader patient community is following here, of really trying to do the necessary work to build the infrastructure to run trials properly and do drug development properly, and to have the patience to get it right. Rushing into trials with therapies that are not really well motivated actually doesn't help anybody. There's all sorts of things that need to be done, including really understanding the diseases where we're running trials, because the only way that we can actually identify the appropriate endpoints is if we really understand the diseases. All of this is really being gotten right in this community. I can hear that from the things that are being said. So I want to start by really acknowledging that, encouraging everyone to keep the faith and keep doing this work the way that it really needs to be done. Next thing I want to say is I just really want to thank specifically the CMT2C community that's gathered here. So it's actually just pointed out to me by Susan, this is really quite a seminal moment. So if you forgive me, I'm going to give a little bit of background to why this is a seminal moment. It's just about now 5 years ago that I started thinking about leaving academia to start a company to develop therapies for some rare diseases. And I started putting together a plan that would include a focus on CMT2C because I felt that it should be a disease where we could develop an effective treatment. And I started talking to investigators to get them to help in this effort. And I will say, for those that don't experience reaching out to important academics about getting them involved in studies, for example, at drug companies and things like that, often important academics will say, well, why don't you talk to me once you have a good drug, and then we can start talking about trials. And it's a little hard to get their attention before you are at the point of having a good drug. So let me call out specifically Brett, who you heard from yesterday and is sitting right over here. He agreed to engage and help. For not only do we not have a drug, we didn't even have a company. So I said, I'm thinking about starting a company. And one of the things we're going to do is we're going to develop a good drug for CMT2C. Now, almost everybody would have said, yeah, right, why don't you call me in a few years? But Brett actually jumped in and started helping right then, and has been helping all the way through, as has Charlotte. So thank you very much, Brett. I also want to really directly acknowledge the CMT2C patients that have come together here today, that Jenny, Claire, and Josephine, and Stephanie. Thank you for being here, for being part of this. This is one of the last times that we'll be coming together before we actually kick off a trial, which is what I was asked to talk about, and I will promise I will shortly get to. But it's really nice that we're here together at this really critically important time. So you heard about TRPV4 and CMT2C from Brett yesterday, but before I give you the update about the trial, let me just briefly highlight how mutations cause disease in this particular form of CMT.
I was always the one.
Before it encodes the calcium channel. We do know that mutations in this channel activate the channel, and we know further that that is actually how you get disease. It happens, as you heard from Brett, through a mechanism that is very, very distinctive, probably different from any other form of CMT, but disease initiates with this overactive channel. What that means is that a precision therapy for this particular form of CMT is a compound that can inhibit that overactive channel. So that addresses the underlying cause of disease. Being able to develop therapies that address underlying causes of disease is the reason that I started Actio, and TRPV4 is the first time that we set out to be able to develop a compound that does that. We believe that we do have a compound that can address the underlying cause of disease. I can't make promises about how it's going to work, obviously, but what I can tell you is that this compound has been designed to be able to inhibit the overactive channel, regardless of what different mutation actually activates the channel. The other thing that I can tell you is that this compound has been tested in the first part of testing a small molecule therapy, which begins by evaluating what happens when you give the compound to what we call healthy volunteers, people that do not have CMT2C or any other known disease. And the questions that you're asking are whether it looks like it's a pretty safe compound and okay to give to patients. And also you're asking, what does the body do to the compound once you put it into a human being? This is something we refer to as PK. And the stuff that we're looking at are things like, well, how long does it stick around? Because if it disappears too quickly, then it can't be there to do its job. And we address other related questions. And all of that is under the label of PK, and it has to have appropriate properties. And what we would ideally like would be appropriate properties to be able to take it once a day, because that would be maximally convenient. So what I can say that emerged from that part of the trial is that we do have a well-tolerated compound. We did not have any important safety issues emerge. And we also have a compound that has appropriate PK characteristics for using it once a day, which means that the patients will be able to, we hope, take this once a day if it ends up actually helping their symptoms, as we very much hope that it does. We'll also say that actually it turned out pretty well in the sense that there wasn't a lot of variability in what happens across people. So all of these things made the compound look really pretty good. The next phase, of course, is to bring this compound to patients. And this is why this is such a seminal moment. That is now what's happening next. This will be done in very close partnership with St. Jude and Johns Hopkins. We are not yet outlining the exact criteria for who will be in the trial, but what I can tell you at a high level is that we will take an inclusive approach to this first trial. We will be looking at a broad range of ages, and we'll be looking at a broad range of presentations. And I would actually just mention, not only is every CMT patient different from the next one. That's even true within subtypes of CMT. So as everyone knows here, there is extreme variability amongst patients with CMT2C, and we will intend to take a fairly inclusive approach to this initial trial. I'm also not permitted to say exactly when it's going to start because there's a lot of variables, but what I can tell you is that we now believe that it's coming fairly soon and that we are hopeful that we'll be starting this before too late into next year. So that's the very quick update from Actio Biosciences in San Diego. The only final thing I want to say is just what a privilege it is for us at the company to really be in a position to be getting this close to hopefully be introducing a therapy that we hope is really going to help. So thanks very much.
And last but not least, we'll have Dr. Karl Bjartmar, Chief Medical Officer from Augustine Therapeutics.
All right, thank you, Maggie. So good afternoon, Karl Bjartmar, MD, PhD, CMO at Augustine. So we are a Belgian company based in Leuven in Belgium, but we have colleagues from different geographies, including the Boston region. But we're a spin-off of the University of Leuven, so that's really where it started some 15 years ago at the lab of Ludwig van der Bosch, and Rob is also in that lab now. And the molecule is a so-called HDAC6 inhibitor. So it's a small molecule that can inhibit an enzyme called HDAC6 selectively, and by doing that, in the lab, it was discovered that could stop the neuropathological progress in a number of models of several diseases. And specifically for CMT, they did a study in a CMT2 mouse model and showing that this molecule did not only stop the progression, but could also reverse the phenotype. So the mice actually got better. So that was in CMT2. And then Augustine took over this molecule and expanded the development. And we also looked into a mouse model of CMT1, CMT1A. And interestingly, we could see the same positive results there. So not only stopping the progression, but also reversing the phenotype. And that was seen in both electrophysiology and also grip strength for this mice. And we looked at the biomarker called neurofilament light. And we could see that biomarker actually reduced, it got better, and it was dose dependent. And then when we looked at safety, this compound molecule in the animals was well tolerated in the tox studies, so it appeared to be safe there. All of this sort of justified moving into to the clinical stage into humans, and that's where we are now. So we're currently doing a phase 1 study, and the phase 1 study is a study in healthy volunteers. With the purpose of finding the right dose levels and also to look for safety and tolerability. And this study started earlier this year, and we expect to finalize the study later this year and have the full picture like early next year. And then if the data allows, then we will bring this into patients. And since we have animal data and the mechanism of action would indicate that there could be potentially a good effect in several genotypes and several subtypes of CMT. The intention is, and I can't be specific at all at this time point, right? But we probably would have like a broad approach when it comes to inclusion criteria. Once we get there, and eventually I could get more specific about it, but that's sort of where we are now, and I think that's what I can say now, right? We're really looking forward to get the final results from the phase 1 study, so we can start planning for the next stage, really moving into the patients. So, thank you.
Thanks so much, Karl. And one final round for everybody.